Hello, this is Group 2, and for our project, we'll be discussing CRISPR technologies and the 2015 moratorium. So, CRISPR is short for clustered interspaced short palindromic repeats, and it describes the genetic appearance of a system. This method functions almost like an immune system used by microbes to protect themselves from harmful viruses. CRISPR establishes a genetic memory and creates a guide RNA sequence to find viruses through the complementary sequence. So that the next time that bacteria infects the cell, RNA will be able to recognize, bind to, and then destroy the sequence. Cas9 is a complex created by two RNAs with a protein. Cas9 is a nuclease or a type of enzyme that can cut DNA. So when a complementary sequence or a guide RNA acquires a target within a viral genome, that DNA can be cut so the virus is then disabled. This same cut and paste system can be engineered to cut DNA at any given location. So Cas9 can be utilized to target and alter human genomes in sperm and egg cells, also referred to as germline cells. It's very important to note that these alterations can be done within a living cell. And in the bigger picture, this provides a unique opportunity to alter the human race. This quickly became very worrisome because moving forward, this allows for targeted genotypical and phenotypical mutations. Thus, a moratorium was called starting in 2015. It's not new to scientists to know how to modify genes. Not only that, but it was formerly a more lengthy, difficult, and expensive process. However, the use of CRISPR and Cas9 technology streamlines gene editing and makes it more accessible. While the possibility to make precise changes to genomes is exciting, it opens the door for editing and prioritizing specific genetic traits such as height, hair color, intelligence, etc. Naturally, grew concerns for creating quote unquote designer babies and the possibility of engineering the human race moving forward. There were divisions among scientists on how to proceed, but a containment strategy was proposed by several groups of scientists and bioethicists to raise awareness for their concerns of these advancements. The intention was to cultivate trust and ideate ways of proceeding judiciously, and this started in 2015. That same year, the National Institute of Health, or the NIH, announced that it would not be funding germline editing on human embryos, and Congress passed the first of many bans of altering germline cells intended for pregnancies. Between 2016 and 2018, national scientific associations, as well as research groups, provided guidelines for genome editing for its other applications, such as in clinical trial treatments or even for the treatment and even removal of disease. And I say disease in quotes, depending on how one defines that. By 2019, this topic had become internationally controversial as a global moratorium was called, and this was following a Chinese scientist or a group of Chinese scientists who had genetically edited embryos, a couple of which were even carried to term. In response, US senators swiftly denounced this and called for the construction of international guidelines. However, at around the same time, some groups began advocating for a partial lift of the ban and changes to restrictions for the use of CRISPR and Cas9 therapies, such to include other, gene therapies such as three-parent IVF. But in that same year, the NIH made its support for a 5-year international moratorium clear and stated that these experiments for clinical purposes were, quote, irresponsible and unethical. Since then, there hasn't been much movement regarding regulation, but the ethical concerns of using CRISPR and Cas9 technology has continued to grow.
Now that we've looked at why the moratorium was introduced, let's talk about one of the biggest ethical questions surrounding CRISPR, and that's consent. So, consent is defined as the voluntary agreement to participate in research with full understanding of its purpose, risks, and benefits. But with CRISPR, this is where things get tricky. Embryos obviously can't give informed consent, and any genetic edits made today could affect future generations, and those generations can't consent to those changes as well. So that means that decisions are being made on behalf of people that don't even exist yet, and that's a real huge ethical responsibility. So it really raises the question of where to draw the line, who gets to decide what's best for the unborn and if we are ready as a society if we're ready to make those kind of decisions. And that's why transparency is so important, and that's why researchers, patients, and the public all need to understand, what's happening and not just the science behind it. So in class, we did talk about fully informed consent and protecting the vulnerable populations. And CRISPR does fit into this discussion because embryos and the future children are the most vulnerable of all. So, all in all, this slide really does remind us that consent isn't just a form that we sign, it's about the communication, honesty, and respect for those that are affected by science. Beyond consent, there's also the question of how society and even religion views CRISPR. Many religions and cultural beliefs view gene editing as interference with nature or divine creation. It raises the moral question of whether humans are crossing the ethical boundaries. So it does challenge the idea of whether humans should be altering life at all. But it's not just a religious issue. Um, People without religious beliefs also have strong views, and they tend to focus more on the social consequences, like fairness, equality, and how this technology might be used. So, an example I can mention is the idea of designer babies, and this is where genes could be chosen or enhanced. Um, I know it sounds like futuristic, but it does seem unfair. So if only the wealthy families in this case could afford these kinds of procedures to be done, that could make the inequality a lot worse. So whether you look at it from a religious, cultural, or just an ethical perspective, the same core question does come up. Um, just because we can, does it mean that we should? So CRISPR does force us to rethink, what's natural, what's moral, and how much control humans should have over evolution. And this does directly lead us into another difficult question. How do we even decide what's considered a problem in our genes to begin with? So this brings me to another point, defining what a faulty gene is. A faulty gene is a gene that carries a mutation which disrupts normal function and may lead to disease or abnormal traits. So CRISPR does give scientists the power to fix or disable these mutations, which does sound great in theory, but it also does open up a big ethical gray area. So it does provoke the question, who decides which traits are undesirable? And where do we draw the line between treating illness and enhancing human ability? An example would be, let's say CRISPR could remove a gene linked to short height. Is that curing some sort of medical condition, or is that simply enhancing someone? It's a fine line, and it's one that's easy to cross without realizing it. So this is where the idea of acting responsibly with limited knowledge comes in. We really don't know or fully understand the long-term effects of gene editing. So once we make that change, it's permanent and there's nothing that we can do to undo it, whether it affects one person or their descendants. So the question I mentioned earlier, who determines which traits are undesirable or worth changing? There's not really one answer or one concrete answer to that. It's something that we as a society, scientists, uh, ethicists, will have to keep discussing as technology develops.
Okay, so now let's look at IRB and research ethics. An IRB, or an institutional review board, does not approve CRISPR as a whole. So instead, the IRB reviews specific protocols in which this CRISPR technology is being used on subjects. So instead, an IRB reviews specific study protocols to ensure that all ethical and regulatory standards are being met. This would include an informed consent, making sure patients understand the research and agree to it voluntarily. This informed consent form is something that's given to the patient, a copy is given to them, and that usually contains a title of the study, the protocol number, a sponsor, which is like the pharmaceutical company sponsoring that, and an investigator. That would be the main PI, the doctor overseeing all sorts of study-related procedures. The IRB also reviews, the risk benefit, ensuring that all potential benefits justify any risk in the study, and subject protections, safeguarding the rights, privacy, and well-being of all research participants. If CRISPR is being used in research involving humans, that particular study must meet IRB approval before any part of that study can begin. In short, an IRB doesn't approve or deny CRISPR as a whole. It just makes sure that any research using it meets the highest ethical standards for human subjects. Privacy and data protection. CRISPR research depends heavily on genomic sequencing data. This information is profoundly personal and identifiable because a person's genome can uniquely identify them, even when it's anonymous, or when the study is blinded, or when it's randomized. This kind of data is extremely sensitive. That's why genetic information is protected under the laws like HIPAA. Here in the United States, we have HIPAA, which safeguards personal health information. Genetic data isn't just scientific material. It's a personal data that reveals, deep insights as to who we are and what makes us who we are. Back in 2015, the CRISPR moratorium highlighted exactly this concern, the need to protect individual privacy before pushing the technology forward. It reminded the scientific community, that before advancing to any powerful tools or technology like CRISPR, we must pause and assess any potential harms. In short, any collection or use of genetic data for CRISPR research raises significant data protection concerns. Protecting individuals' privacy isn't just an ethical requirement, it's essential to maintaining the trust between the subject and the research, and overall important to maintain that integrity in research. What now? As we've been discussing, this CRISPR technology has raised a lot of scientific and ethical questions. This germline editing, that's again editing of DNA of embryos in a way that can be passed down to future generations. So it is hereditary. Anything that's edited will be passed down. It's hereditary. But of course, there was someone who pushed these boundaries. So in 2018, a Chinese scientist, I hope I don't butcher this really bad, his name is He Jiankyu. So he broke this international norm by using CRISPR to edit human embryos. And because of this, these two embryos developed into twin girls. So they are the first humans ever with intentional edited genes. What the scientist was trying to achieve, he targeted this specific gene called CCR5. For my understanding, it's what affects how HIV enters the cells. So by changing this gene, his goal was to make these twin girls resistant to HIV. However, the CCR5 gene also plays other roles into the immune system, not just for HIV. So tampering with this gene, could make it's unpredictable what side effects could these twin girls potentially have. This experiment, of course, called for global outrage. Scientists and ethicists around the world condemned the actions of the scientists, calling it, unethical and reckless. Since then, many countries have banned and heavily restricted this germline editing. This field has hit a pause for now, but as science and technology continue to advance, the question remains, when is it right to edit life? Should we even edit life itself? In conclusion, the 2015 CRISPR moratorium, it refers to a temporary pause that scientists called for this CRISPR technology on human editing. This obviously has raised several ethical questions. For example, should scientists be allowed to edit human germline at all? How should we determine when it's safe enough to proceed? How does this quote unquote hold impede life-saving research? That's a question from like the other side of the coin. How should society prevent any inequality in the enhancement of, if this came to be moving forward, how would we prevent that? Therapy and enhancement, when do we draw the line? Research is not always just black and white. There's a lot of gray area in there. And can we ever reach a consensus on gene editing? Can that ever be achieved? So there's a lot of heavy ethical questions regarding all of this, and it's what made us gravitate towards this specific subject. So we really hope you guys enjoyed this and hope you found it a little bit interesting, just like we did. If you have any questions, we'll go ahead and answer them in the thread from the Brightspace. Thank you so much for listening in.